Okay, we're going to go ahead and we're going to start with this supergroup first. Again, I'm not really doing this in any particular order. I'm going to keep this biggest one for the next lecture. So we'll start here with Archaeplastida. So if you look at the term Archaeplastida, the archae looks like the archaea, right? And that just means archaic or old. And the plastida here refers to the fact that the cells have plastids. And a plastid is a type of organelle that stores pigments. And that means that the organism then usually is going to be autotrophic. So plastid, maybe you've heard of a chloroplast before. And a chloroplast is an organelle that stores chlorophyll, green pigment, hence the term chloroplast, okay? And so the organisms that fall under this, and they've been around a while, obviously, right, because of the prefix here, archae, we're going to have clade Rhodophyta. So the supergroup is Archaeplastida. It's broken down into 3 smaller clades: Rhodophyta, Chlorophyta, and Charophyta. The Rhodophyta here, this is silly, but I try to remember Rhodophyta those are the red algae. Chlorophyta, that sounds a whole lot like chlorophyll, right? So those are going to be your green algae. And then the last clade here, Charophyta, these are the organisms that are thought to be the most common ancestor to land plants. And we're just going to mention those and move on. We'll talk about these in a little bit more detail when we move over to the plant. So right now I'm going to focus on Rhodophyta and Chlorophyta. Okay, so let's look at the red algae first. And that's going to take us, guys, to the top of page 4 here. C, survey of the supergroups. 1, supergroup Archaeplastida. A, clade Rhodophyta, the red algae. Okay, so I'm sure that you can imagine, right? Because they're red, they've got pigments, they're going to be able to undergo photosynthesis, so they are autotrophic. And they look red because they have more phycoerythrin, which is the red pigment, than they have chlorophyll. Most of these guys are going to be multicellular. So as you can see, there's branching, right? And then branching here. They look like little mini plants, but they're not true plants, guys, because they don't have tissues and organs. Remember, they are as complex as just being multicellular. They are fairly delicate, and so what that means is that if you were to go try to find some rhodophytes in the ocean, you would need to go down into some of the deeper waters, because down in the deeper depths of the ocean, they're not going to be subjected to waves. And because they're so delicate, any waves that would come by would just basically destroy the organisms. And so they're found at deeper depths. And that works for them because of the phycoerythrin. That means that they are able to gather sunlight, UV rays of sunlight at some of the deeper depths of the ocean. Very different light reaches those areas of the ocean than, like, let's say, the top of the ocean. You might be really familiar with it and maybe not even know it if you are a sushi eater, right? If you're a fan of sushi, that really dark, kind of almost black that we have going around these rolls here, that is made up of a specific type of red algae called nori. And that's going to be that paper-like right there. And these guys are going to be the most abundant of all algae in the tropical waters. Again, down deeper in the part of the ocean where they can get the proper UV light for the red pigment phycoerythrin to capture for photosynthesis, because they are autotrophic. And then it protects them because they are very, very fragile. Okay, the next group of algae that we have to look at, these are going to be your green algae. So I'm still in the same supergroup, Archaeplastida, and I tried to color code these guys as best as I could. The clade now, though, instead of being Rhodophyta, is called Chlorophyta, and these are going to be your green algae. Okay, they are found in many more places than the red algae. They are found in freshwaters, they are found in marine types of habitats, and they are found even growing on terrestrial type of environments, even at some of the highest elevations. A couple years ago, we went on a hike in Mammoth, and it was in the middle of summer, but there was still some snow on the ground because we were at an elevation of almost 10,000 feet there. And there were little pockets of kind of this green glowing snow, and somebody mentioned it, and we all looked over there, and they wanted to go look at it and try to eat it to see if it had any taste. They were being silly. We said, no, no, don't do that because of the whole algal bloom, right, and the carcinogens. But they can live in very high altitudes because they have special proteins that allow their cells to handle these intense UV types of lights that you're going to get at the very high elevation. So a lot more variation in variety here in this group than you have in Rhodophyta. Okay. You have some of these that are really important as symbiotic organisms. The sea anemone example that I just gave as an example at the beginning of the lecture. Remember the single green algal cell living inside of the little tentacles of the coral polyp. And then another example of a symbiotic type of chlorophyte would be, again, single cell green algal cells that live in fungus, and those are called lichens. And when we get to the lecture on the fungi, I'll show you that. But really important because primarily they're getting a house, and in return they're giving off sugars. Okay, so it's just as simple as that. Okay, two organisms that I want to highlight here that you guys are going to be looking at in the lab that are in Chlorophyta are Volvox and then this other organism called Spirogyra. So I highlighted this one already earlier, but again, this is daughter colonies that are inside the parent colony. So this is the parent colony, and then all of the smaller little beach balls, those are going to be your daughter colonies, and this is all due to asexual reproduction. But I wanted to show you, using this diagram over here, a little bit of the detail that you have going on here that you wouldn't be able to make out even if we were in the lab looking under the slide. But this parent colony right here, guys, okay, the outermost barrier, if you will, is made up of many unicellular organisms just growing together. And these unicellular organisms look like this. They have a little red eye spot, and that helps them to detect light. And then they are flagellated, so they have two flagella. And so when you look at this in real life, these things kind of spin around in the environment, and they kind of float very peacefully through the water. And they're able to do that because all of these organisms here are beating their flagella in unison and in the same direction, because they're trying to get to the light, which again is being detected then by the red eye spots. Okay. And then any of the other terms that are on here, you are not responsible for. Okay, so there is a, hold on here, guys, sorry. Let me just go through, we'll just do it this way. A YouTube video here. Let me see if I can get it to pull up. There we go. Okay, sorry, just be patient. I'm still here. And this is just going to show, maybe, it's not. What I'm trying to show you is a Volvox moving in the water. This is one of the organisms that we actually ordered to have living in the lab so that you could have made a wet mount. Okay, we're not going to wait for that anymore. Okay, so we'll just ignore the hyperlinks. If you want to, you guys can go and try to do these little YouTube videos on your own. After you're done listening to this. So I'm going to have to kind of work out those kinks there. So for that, I apologize. Okay, so the second example, another example that you'll be looking at and having to draw in the lab for the Protist, then another member of Chlorophyta, then this one here is called Spirogyra. And this one looks very different than the Volvox. This one, instead of it being a round colony, this is referred to as a filamentous green algae. is you can kind of make out there's a cell wall there, there's a cell wall there. So that's one organism connected to another organism, so on and so forth, making a chain, which is a filament. And then the most obvious part of this that I want you guys to highlight, they're kind of like these little X's, but they're actually little spirals. And these spirals, then, those are the chloroplasts. Okay, so lots and lots and lots of chloroplasts with lots of chlorophyll, then allowing for photosynthesis to occur. Okay, so one of the important things for this group, then, is the type of reproduction that they do. And so they can break off from one another. The chains can from the filaments, and they can continue to grow via asexual reproduction. But they can also grow using sexual reproduction. And so this is going to be called conjugation, and this is a term that I did use in the last lecture to explain when some types of bacteria undergo sex, if you will. They use conjugation. That's one of the methods that is available to them. This is similar, but just a little bit different. So if we look here, what's happening is that we have two filaments that are coming side by side. So there's one filament with its chloroplast and another filament with its chloroplast. And when they align, you're going to start to see then a little bulging between each of the two cells within the filament that are here. And these are called conjugation tubes. And these tubes, kind of like a sex pilus, right, is going to allow for the genetic information to move from one cell to the next. And so if I go over here, and then in the third step of the process, then the conjugation tube has fully developed and completely open, connecting that cytoplasm to that cytoplasm, same here and there. And you can see then that the entire cellular content is moving from this cell to that cell, same here and same there. So you could think of this as the quote-unquote male, if you will, and then the quote-unquote female. So here it's empty because all of the male material moved over to the opposite side. All of the information, including the genetics, was combined, right? Then giving us what are called zygotes. So think of the sperm and an egg cell and animals coming together to give a zygote. That's the same thing that has happened here. And then what will happen is that these will go through mitosis and they will make more cells via asexual reproduction to resume the growth of the filaments. Okay, another hyperlink there that if you'd like to go watch on your own, please do that. What it is, it's just showing that the male information here, the male gamete, is going to literally move through the conjugation tube into the female, and a zygote forms. And so that shows you in real time. So please go take a peek at that if you can. Okay, so let's see, that takes care of my slides, at least for Archaeplastida. But if you're in your notes here following along, see number 1, part C, clade Charophyta. I do not have a slide for this. And again, this is just the group of organisms that are thought to be the common ancestor to land plants. So the plants that you know today that are alive and well probably evolved from these guys here. And that's all that I'll say about that now. We'll come and take a peek at that later when we get to our plants. Okay, so that means that we can move on, and we're going to go then and look at the next supergroup, which is the supergroup Excavata. Okay, so for this supergroup Excavata, the name comes from the fact that they all have what's called an excavated feeding groove in their cell. And if you excavate something, you're like kind of cutting it out. So it's just a space somewhere in the cell, maybe under the membrane, for example, that is going to act as a feeding groove, quote unquote, a mouth. And so that's the common characteristic that we see in this group here. Okay, so we have 3 clades, Diplomonadida, we have the clade Parabacilla, and then we have the clade Excavata. And Excavata, the clade Excavata is broken down into 2 subclades. So these are our first subclades that we're looking at. So the clade Excavata is broken down further into the subclade Kinetoplastida and then the subclade Euglenoophyta. Okay, so we're going to go ahead and look at the diplomonads first. Okay, So what you've got going on here, guys, is that these are very different than what we just looked at with the Archaeoplastida. But these guys here, for the diplomonads, they are anaerobic. And if you remember from the lecture on the prokaryotes, anaerobic means, right, that they live without oxygen. And these guys have to be in an area that does not have oxygen. Oxygen actually poisons them. Couple of kind of crazy things about these guys is they have 2 equal-sized nuclei. So there they are. They kind of look like eyes, right? And 2 equal-sized nuclei there. And that's kind of an unusual characteristic seen in only a couple of these protists, this being one of them. They do not have true mitochondria like you and I do. And I know we didn't go over the organelle, so let me just do that really quickly with you now. A mitochondrion is an organelle found in all eukaryotes, or I should say most eukaryotes, that breaks down sugar and makes energy for your cells. And they don't really truly understand why the organisms in this clade are lacking mitochondria. But in place of mitochondria, they have these structures called mitosomes. And a mitosome, think of it as a very small kind of stripped down mitochondria. So its function is the same. It is for cellular respiration to take energy and break it down into a usable form of energy for your cells. And their best guess at this point is that they have mitosomes because they got those mitosomes from their ancestors. And then that, interestingly enough, begs the question, are mitochondria even necessary? So if these guys here can live with these very simple structures called mitosomes and have enough energy to stay alive, well, could the rest of us? But just a question just out of curiosity more than anything. Okay, you can see here that they have multiple flagella here and here, and then on the picture they're showing the same thing. And then our representative for the group is called Giardia. That is the name of the genus here. And this guy, I think it's kind of funny because you see this hiker over here. It's thirsty. He's thirsty. He's, you know, maybe up in the sequoias, and you've got a creek or a river that's running quickly, and it looks like it's clean, and well, no harm, no foul, right? I'll go ahead and just take some of this water here. They supposedly bottle water straight from the source. They don't. You don't want to do this because a lot of our waters, especially here in California, have this guy, Giardia. And it looks like it's grinning at you, right, as it's making its way into your mouth and then down into the intestine, where it's going to have a great time, but you are not. And so if you look here at this next slide, this is showing then intestinal tissue, which is all of this stuff here. And every single one of these guys then is an organism of Giardia. And so you can see that they overpopulate themselves, and they're going to live off of the nutrients and basically destroy the cells that are lining your intestine there. And you're going to be very sick. It can lead to things like amoebic dysentery, which is a very, very tough type of diarrhea, for example, and can lead to dehydration. Okay, next, following right along then, same supergroup as excavata, but now we are in the clade Parabacilla. And one thing that you want to know about this is, like the Giardia and the diplomonads, is that these two are also anaerobic, so they're going to be found living in organisms that or environments that lack oxygen. They don't have any nuclei, so that's kind of a unique characteristic. Instead of having the true mitochondria, they'll also have mitosomes. And then these structures here, number 3, called hydrogenosomes. And a hydrogenosome is a novel, kind of unique organelle that these organisms have. And a hydrogenosome releases hydrogen peroxide. And they release hydrogen peroxide in their area. And what that does, think of the hydrogen peroxide that you have maybe in your cabinet, right, in the bathroom. You go and you fall while you're outside and you scrape yourself, and you go get that brown bottle of hydrogen peroxide and you put that over the wound, right? And maybe it stings a little bit, but why are you doing that? You're doing that because you want to kill any of the superficial bacteria that's there that now has gained entry into the body. And so that's kind of the same thing that's happening here, is that this organism, from their hydrogenosomes, they release hydrogen peroxide. What that does is it changes the pH of the area, then killing the good bacteria. So it's able to outcompete the bacteria, and it now has full access to the environment. And in this case, it would be the lining of the vaginal canal. This one specifically here is called Trichomonas vaginalis, and it is a sexually transmitted infection that are found, again, like I said, in the vaginal canal. So they are able to move around because they have these kind of ribbon-like structures here, and then there's another one right here. These are called undulating membranes, allow for local motion, in addition to their flagella, okay, that allow for local motion. You guys in your lab are going to be asked to look at a micrograph similar to this one right here and to draw it. I like to show this picture here. This is actually from an electron microscope, magnified way more than we could ever get, but I like to kind of give you a heads up as to what it is that you're supposed to be finding. So these little guys right here. So that would be the undulating membrane, and then these are all of the flagella. Okay. Okay, fun stuff, right? Okay, next group is the clade Euglenozoa, and again, we have two subclades here. Before I get to talking about the subclades, the common characteristic for the clade Euglenozoa is that these organisms have a flagella, which is not anything new, but the flagella has a crystalline rod for structural support. Not that you would ever be able to see that, guys. I'm just trying to point that out to you. Okay, any organism within this clade can either be heterotrophic, they can be autotrophic, or they can be mixotrophic, and that's a new term that I haven't used yet. So I'm looking at C, number 2, extreme range of metabolic diversity for Euglenozoa. So go ahead in your notes just off to the side and write the term mixotroph. It's spelled M-I-X-O-T-R-O-P-H. And it does exactly as the name implies. It's able to mix the type of feeding that it does. So if it is autotrophic, like this guy right here, and it has the available light source because it has the green pigment, it'll be autotrophic. But if it finds itself in an environment that is lacking light, then instead of dying, what they do is they turn on different types of feeding mechanisms and they become heterotrophic. And they become predators, for example, to things like bacteria and other smaller protists. Not all of the organisms in this group can do that. Euglena here on the right is one of them that can. So I'll try to point that out as we work our way through. Okay, so let's go ahead and look at the first subclade then, called Kinetoplastida. And if I'm looking at this guy right here, subclade is Kinetoplastida. And the representative organism that you'll be looking at in the lab is called Trypanosoma. And Trypanosoma is this organism here. There's another one there and another one there, kind of like these little purpley worm structures. And they are referred to as a hemoflagellate because they live then inside with your red blood cells. Probably not a good idea, right, to have this type of protist living inside your blood in amongst your red blood cells. They prefer to be in the plasma, so that's the liquidy part of your blood. And this is the disease that's caused by this is called African sleeping sickness, and it is vectored by the tsetse fly. Very, very rare though. There's about 1,000 cases a year in the most rural parts of Africa. And it's called African sleeping sickness because it's basically the person doesn't wake up one day. But they are sick and they do have symptoms and signs leading up to that point. Usually it starts with a fever and a headache. The person might experience very achy joints. They can become very confused because this can lead to problems within the nervous system, and that would be an advanced stage then when the person then might just not wake up. So sleeping sickness, sometimes called African sleeping sickness, is caused rather by that protist. Okay, the next subclade, this one for whatever reason is very similar to the name of the clade, but the subclade here is Euglenophyta. Guess what I want to point out is anytime you see the prefix phyta, that refers to plant. And so if it refers to plant, then it's safe to assume in most cases that it's plant-like and therefore is probably going to be autotrophic, which this guy is, except it happens to be also a mixotroph. So this right here is Euglena. If you look at a slide of Euglena, this is very magnified. And so the Euglena on your slide would basically, you wouldn't be able to see any of the internal contents of the cell. It would look kind of just like a little tiny canoe, and you might be able to make out the nucleus, okay, which in this case it's not stained, but it would be stained in your slide, the nucleus. They have two flagella. Again, you would never see that. Let me come over here and point that out. A very short flagellum and then this long flagellum. They too, like the volvox in the Chlorophyta, have a red eye spot, and the red eye spot allows them to detect light in addition to their light detector. Okay, something that I want to point out that we haven't seen at all, let me introduce it to you here, is going to be this organelle called a contractile vacuole. And so it's this structure right here, this organism right here, kind of like a little pouch or a sac that's inside the cytoplasm. And what the contractile vacuole does, guys, is this is going to basically control the water or control osmosis, water coming in and going out of the cell. Because if you're living in water, by the way, you should be able to pick up on that because it's flagellated and the flagella only work in water. But if you're living in the water, do you think that there's a possibility that you could drown yourself? And the answer is yes. And so that's the job of the contractile vacuole then, is to control the amount of water that's inside the cell so that the organism does not literally drown itself. Nucleus, that's where the DNA is stored. The chloroplast is for photosynthesis, and then the plasma membrane, no cell wall. Don't worry about the pellicle here. That's just for structure, but I won't ever ask you anything about that. Okay, we're going to move on then and finish up. Our last supergroup is the Uniconta group. So this is at the top of page 5. And you have one clade, amoebozoa, but two subclades, gymna amoeba and enta-amoeba. And so little tricks, right, of the trade to try to remember who's who and what's what. Amoebozoa is the clade, and amoeba, that term, is in the subclade, each of the subclade names. Okay, for the organisms here, for the Uniconta, they are going to have what are called lobed or tube-shaped pseudopods. Pseudopod, P-S-E-U-D-O-P-O-D. Pseudo means false or fake, and pod, guys, means foot. So think about it. If you're going to go to the podiatrist, right, that's somebody that's going to look at your feet. So they have the common characteristic for the group here is lobe-shaped pseudopods, and I will show you a picture of those here in just a minute. Okay, so let's look at the first subclade, gymna amoeba. And these are going to be organisms that you may have heard of before, just the regular old amoeba, and this specific one is called amoeba Protist. And these have very fat or lobe-shaped pseudopods, which is going to be that guy, and then there's another one there, and then there's another one there, for example. That would be the nucleus, and then kind of all of this granular stuff. Those are all of the structures, right? All of the organelles that are inside the cytoplasm of the cell. We find these guys living in soil. They're found in fresh water and also in marine type of environments. And these guys on this side of the screen are going to be free living, which means that they do not require a host. So they're not typically pathogenic or parasitic or anything like that. They just eat off dead, decaying organic matter. They can have a cyst stage in this group, and that would be over here for this guy. So I'm going to kind of just jump over here. And the subclade enta-amoeba, these are amoeba that you probably want to stay away from, specifically this one here, enta-amoeba histolytica. And so this one here, the cyst stage, so I've jumped down to the subclade enta-amoeba in your notes. Cyst stage is a hard outer wall that's going to allow for overwintering, kind of very similar to the cyst stage that we talked about when we did our brine shrimp experiment, okay? And that's going to allow for protection of the organism when it's in an area that it's not liking. this is something that maybe if you go overseas or you go to a different country and they say don't drink the water, or don't, eat a salad because maybe the produce has been washed with water, and that's water that is not part of your normal flora in your gut. So you accidentally ingest this, and kind of like the Giardia, it's going to have the same effect. It's going to go in and it's actually going to infect your body, and you're not going to feel good. I'll come back to that one in a second. But right here, if we go to the life cycle of the Entamoeba histolytica, so that cyst stage that I was just showing, you somehow ingest it. It becomes metabolically active, okay? And then it grows. And so these cells are going to get bigger, and they're going to migrate, and they're also reproduce themselves asexually. They're going to make their way into your intestine, move through the body from there into the bloodstream, because they can cross then membranes. They can make their way to the liver, the brain, and the lungs, and that would be worst case scenario. As that part of the life cycle is happening, then they can also make more cyst stages for themselves, and that cyst stage then is going to exit the organism, in this case a human, out of the stool, maybe into the water, and then the whole cycle repeats itself in the second person, or the second host that is hospitable. Okay, one last thing. I'm going to just back up for just a second, guys. I apologize for that, but hopefully you can follow along here. This right here is just a drawing, a very generic drawing of an amoeba, more like a gymna amoeba. One that wouldn't hurt you, but I just wanted you to see that there are nice labels there. And something that we've seen just before, there's your contractile vacuole to control the amount of water, but now we've got food vacuoles. And these food vacuoles are going to take in the food, and they're going to digest it, because these guys don't have a gut, right? They don't have a digestive tract like you and I do. It's a single cell, for God's sake. So how in the world are they going to actually digest their food? Well, they're going to use these specific organelles called food vacuoles to do just that. Now, I do want to see if I can show you this guy here and here. And I'm going to play this one here is an amoeba eating. Okay, that's not going to work either. All right, so definitely some kinks to work out, but maybe again you can go, oh, just kidding. Okay. So if it'll play for me, let me at least explain to you, so maybe if you have to go watch it on your own, you can do so. So the way that they make these pseudopods, these false feet, is by what is called cytoplasmic streaming. So all of the inner part of the cell, that's the cytoplasm, is going to move and stream and push itself, causing these false feet. And they're going to move in the direction as to where the food is. And I really wish that this would show so that you understand. I think it's buffering here. Okay. So if you can go and you can play this, you can kind of see when it lets us, right? The cytoplasm is pushing. It's pushing against a plasma membrane. There's a piece of food right there. It's probably another type of protist. And that right there and that right there, those are two pseudopods, and they're going to come together, and they're going to kind of like give the food right here a big hug. And when they do that, they have actually performed phagocytosis. So by surrounding them with their pseudopods, so surrounding the food right here with the pseudopods, they've now completely engulfed that food, and that food is in the cytoplasm. And you can see here, if it goes, that this little guy right here is going to try its hardest to escape. And you can't really see it very well there, but it's trying to move, but it won't, and it will eventually die. And then the food vacuoles will come in, and they'll basically digest that food that we have going on there. Okay. All right, so we have a black screen now. There you go. We end with this here. Actually, with the CD, with the into amoeba. And I'm not going to be able to get that right now, guys. So anyways, that's okay because I got through all of the notes anyways. So the last slide here that I did show you. Let me see if I can move my mouse. There we go. Okay. Is the cause of amoebic dysentery, and amoebic dysentery causes about 1,000 deaths per year. There we go. Finally came up. 100,000 deaths per year, because why this is such a big deal here is it causes massive dehydration. And you can die from that within a couple of days. Okay, that's the end of that. I'm sorry for the little glitches that we've got going on there. Again, I'm going to practice and try to get through that so that it works a little bit better next time. Okay, email me if you have any questions about the material, and until next time, thank you very much.